Yeah, so today in Bordeaux, the temperature reached 36, tomorrow 38, and Friday more than 41 degrees Celsius. So the highest temperature since records began. So I think it's the right day to talk about climate change. This work was conducted at ISVV with the support of Seguin-Moreau cooperage. And this work started in 2011, just after two warm vintages in Bordeaux, 2009 and 2010, with considerable consequences in terms of aromatic balance. The intrinsic quality of a wine is strongly linked with its volatile compound composition, as previously described by Philippe, involved in the complexity of wine's subtle nuances. Nowadays, it is well accepted that macroscopic effect of climate change can modify the environmental condition of grape growing at global scale in all vineyards worldwide. To illustrate this, I would like to share with you today some data obtained from my colleague Greg Gambetta at ISVV and published recently in DNO. Here it reports several, different temperature metrics from the 50s to 2020 in Napa and also in Bordeaux in red. As you can see here, they all support the same conclusion. Temperatures have increased significantly during these last decades. On the right-hand side of this slide is depicted the historical changes in the average growing season temperature from 1st of April to end of October. It is interesting to note that nowadays, the temperature in Bordeaux are very similar to what's observed in Napa. On the left-hand side of this slide, you can observe the average maximum temperature, and this average maximum temperature increase also both in Napa and in Bordeaux. But it is interesting to observe that the rate is faster in Bordeaux compared to Napa. Another interesting parameter to take into account is the number of heat spike day, defined either a day with a temperature higher than 34 degrees Celsius. Here, I report the number of days, the comparison between this data between Bordeaux and also Napa. What we can observe is that there is a clear increase both in Napa and in Bordeaux. And this increase has very probably a strong impact on the final quality of the grapes composition, of course, but also on the wine quality. So, macroscopic effect of climate change modify the environmental condition of grape growing at local scale, as depicted on this slide. For example, one of the main effects observed a long time ago, it was in Saint-Emilion, concerned the impact of climate change on the ethanol concentration and also on the pH evolution. As you can see, there is a clear increase of the ethanol concentration and at the same time of the pH. So this increase is linked with the higher maturity of the grapes and the ripening condition during the maturation. What is important to keep in mind is that maturation has a strong impact on the aroma of the young red wine, from bell pepper to blackcurrant, blackberry, and figs or prune when grapes are harvested lately. And it is interesting to observe that more and more the frequency of this aroma are more and more found in, especially in Merlot wine in Bordeaux. That's why it's important also to keep in mind that the impact of the maturation condition will modify the aroma of the young wine, and this modification of the aroma will impact what we call the long-term consumption of red wine, as we have already observed with my colleague Sophie Temper, and also impacts what we call the willingness to pay. And also this modification of the aroma of the young wine impact also what we call typicality, as defined previously by Philippe, impact the typicality, and at the end induce indirect financial loss. That's why it's important to understand this phenomenon for a better control and, of course, at the end, management. So the project I would like to describe today concerns the identification of chemical markers responsible for dried fruit aromas in grapes and wines. Of course, it concerns development of a methodology for the quantification of aroma compound. And at the end, the goal is to evaluate agronomical and enological parameters triggering the formation of this compound. So the first step of this project was to select must and wines and red wines marked or not by dried fruit aromas. And to identify these volatile markers, we use traditional gas chromatography coupled with different detectors. And thanks to this approach, we identified several aroma compounds belonging to different chemical families, including furanones, ketone, and lactone. And also, we developed a specific quantization method for the quantization in must and also in wine. The first experiment following the identification task was conducted in order to answer the question concerning the link between the aroma of the must and the aroma of the wine. The question was, is it possible to predict the aroma of must, or the aroma of wine, thanks to the analysis of the must? To do that, we selected several grape samples from 2012 and 2014 vintages with Merlot and Cabernet, with Merlot and Cabernet Sauvignon. Of course, we selected several harvest dates, and microvinification were conducted at the lab. At the end of the fermentation process, samples were analyzed, and we asked a professional trained judge to evaluate several descriptors on the 0 to 7 scale. So I'd like to share some results with you today. So what we observe is concerning the bell pepper green aromas is that there is a correlation, a low correlation, but significant correlation between the aroma of the must and the aroma of the wine. Concerning the fresh fruity aromas, there is no correlation between the intensity detected in must and the intensity detected in wine. At the end, it's not surprising when we know that this aroma is strongly linked with the fermentation process. So now, concerning the aroma, the dried fruit aroma detected, the intensity of the dried fruit aroma detected in must and the intensity detected in wine. Here we can observe there is a quite good correlation between the intensity detected in must and the intensity detected in wine. It means that it is possible to predict the aroma of the wine thanks to the sensory evaluation of the must. So now we selected some chemical markers and we analyzed some chemical markers involved in the dried fruit flavor aroma. I would like to show you some results we obtained in order to illustrate the impact of over-ripening on the evolution of the aroma and the evolution of the chemical composition of the wine. So I propose first to show you some results concerning two high-impact compounds. The first one is furaniol, reminiscent of strawberry candy, and gamma-nonalactone, reminiscent of cooked peach and cooked apricot. Here I compare the results obtained in different samples in must and wine, marked or not by dried fruit aromas. Here we can observe that there is a significant differences were detected in samples marked or not by dried fruit aromas for fruaniol, but on the contrary, these differences were not so clear for gamma-nonalactone. Now, when we observe the results obtained in wine, we can, concerning fruaniols, there is a very clear increase of the concentration of this compound in the wine. Concerning gamma-nonalactone, we observe a similar evolution. So it means that there is a very good link between what we smell in terms of dried fruit, cooked fruit, and what we are able to assay thanks to, specific technical stuff. So now the question is, thanks to this compound, are we able to mimic what we are able to smell? It means when we add this compound in a red wine non-marked by dried fruit aromas, are we able to mimic the dried fruit aromas detected in wine elaborated with overripe grapes? To answer this question, we conducted a very simple experiment, but very efficient. We add simultaneously several different compounds, including gamma-nonalactone and fruaniol. And what we observe is what we add this compound together, we are able to increase significantly the intensity of the dried fruit aromas. It means that these compounds are involved in the dried fruit aromas of must and wine, and we are able to mimic these specific nuances detected in wines. So now I would like to share with you some results obtained. To illustrate all these results, I would like to share with you some results obtained a long time ago concerning the impact of harvest date on the evolution of the aroma of the wine and also on the evolution of the specific chemicals. And I selected today a compound called gamma-nonalactone presented previously. Here, a sample plot located in Pauillac was harvested at different dates, from D1 to D4. On this slide, we can observe that, there is no clear differences concerning the reducing sugar level, the pH, and the total acidity, and even malic acid. It means that thanks to the quantitation of these parameters, analytical parameters, we are not able to discriminate, to differentiate the impact of harvest date on the composition of the must and at the end on the wine. So what is the impact on the fine composition of the wine? So grapes were harvested, and after the vinification, the fermentation, wines were evaluated. And we observe a clear effect of the harvest date, a decrease in the intensity of the vegetal character, and at the same time, an increase in the dried fruit aroma intensity. When we determine gamma-nonalactone, we can observe that there is a clear impact of the harvest date on the gamma-nonalactone detected specifically in wines. It means that, as described previously, this compound is a quite good marker of the harvest date and the maturity of the grapes. It's important to say that we did the same experiments with Cabernet Sauvignon, and we observed the same results. But what is very interesting to note is that the concentration obtained at the end were, I mean, very low compared to those obtained in Merlot. And we will see later how it works, in fact, in terms of ripening phenomenon. In the next 3 slides, I would like to share with you some results obtained and concerning the distribution of this compound in old wines. We know that the maturity impacts the concentration of this compound in the young wine. So how it works during aging. So we can we determine this compound here in 2 groves located in Pomerol and Lalande de Pomerol in the Bordeaux area. We can observe that, I mean, in general, the concentration are quite low, below the detection threshold, which is 26 microgram per liter. The concentration are low, except for one vintage, the 2003 vintage, where concentration are, I mean, very high, 63 and more than 100 microgram per liters. So I remind you that, I mean, for Bordeaux, in Bordeaux, the 2003 was very hot, not so dry, but very, very hot. And now we did the same thing, but with wine samples from Napa Valley, where the climate is completely different, I mean, compared to Bordeaux. We selected two wineries in Napa, located in Rutherford and in the Forth Valley at higher altitude. And what we observe is the concentration of this compound are very different according to the growth, and in general higher for the growth in Rutherford, so at lower altitude. It means that very probably there is an impact of climatic condition, the altitude, and very probably also an impact of the harvest date on the Merlot wine aroma compound composition. So now, if we compare the value obtained with the value obtained for a Cabernet Sauvignon, a great Cabernet Sauvignon wine, also in Napa, it's very interesting to compare the result we obtained for Merlot wines and for Cabernet Sauvignon wine. Here, we compare the distribution of gamma-nonylactone in the same winery, for the same wineries located in Napa, for the 2001 and 2015 range. It's once again, it's very interesting to note that the concentration are quite different according to the vintage. We can observe 2 extreme vintages in terms of concentration, 2010 and 2007, and the other extreme, the lowest concentration, was 2011. And it's very interesting because when you know that the 2007 was very hot, and the grapes in Napa, they had a strong heat waves 2 weeks before harvesting the grapes. So it's explained the highest concentration observed. And on the contrary, 2011 was an unusual vintage for Napa, with very wet and not cold, but not so warm. And that's why we can observe very low concentration of this compound. So gamma-nonylactone at the end is a marker of the maturity of the grape. It's a marker of the climate stress, climate stress. So to go further, we studied the dehydration and the impact of the light on the formation of this lactone for both Merlot and Cabernet Sauvignon grapes. We conducted this experiment in a room with controlled temperature and controlled relative humidity and also light. Here we can observe that for must, when grapes are dehydrated, we can observe a small increase in gamma non-lactone concentration for Merlot, but nothing or almost nothing for Cabernet Sauvignon. Now in wine, we can observe a clear increase in this compound for grapes dehydrated with light. And this phenomenon was also observed for Cabernet Sauvignon, but in a lower extent. It means that Merlot is more sensitive compared to Cabernet Sauvignon. It means that Merlot is able to contain higher gamma non-lactone level compared to Cabernet. And it confirms what we observed previously in wine from Bordeaux and from Napa Valley. So to go further, we try to understand and to identify chemical pattern explaining the formation of this compound in wine. And thanks to a study conducted at ISVV with my colleagues Cécile Thibon and Svetlana Poix, we identified for the first time a new precursor of this lactone. This compound is called 4-oxoanoic acid. And when we spike a must with this compound here in green, we can observe that during alcoholic fermentations, there is a decrease in its concentration and at the same time, an increase in the gamma non-lactone concentration here in red. We confirm this result using labeled gamma non-lactone and labeled precursor for oxoanoic acid. And we observe the same result. It means that there is a direct link between these two compounds, and the Saccharomyces cerevisiae has a strong impact on the formation of this compound. Now the question is it possible to identify a link between the concentration of this precursor detected in must and the concentration of gamma non-lactone detected in wine? And we demonstrate this thanks to the quantitation of this compound in several musts. And after alcoholic fermentation, we can show that there is a quite good link between these two parameters. It means that 4-oxoanoic acid detected in grapes is a direct precursor of gamma non-lactone in wines. So it means that it is, at the end, possible to predict the aroma of the young wine and the old wine thanks to the analysis of this compound directly in the must. So to conclude this presentation, so now you know that we have some key compounds, markers of dried fruit aromas in must and red wines. We demonstrated that harvest date has a strong effect on the aroma and at the end, the composition of must and wines. And our research can help the winemaker because today it is able to modulate the impact of climate condition on the quality of this wine thanks to the quantitation of specific compounds. And at the end, it is possible for the winemaker to predict part of the aroma of the young wine and at the end of the old red wines, thanks to the assay of specific key compounds it must. And also, I think that this research is important because this research can contribute to the selection of new varieties more resistant to drought and temperature with a lower ability to produce dried fruit aromas. So thank you for your attention.